In this short lecture, what we're going to do is review the process of the central dogma. You may recall that the central dogma describes flow of genetic information from DNA to RNA to protein. When we looked at this model by analogy, we talked about how DNA is like a book, and that book is locked in the special location region of the library. Well, that information somehow has to get out of the nucleus, and so that form that it comes out as is the RNA. And in fact, the RNA polymerase is the enzyme that functions like a scribe to take DNA and to transcribe it into an RNA sequence. We said that the RNA is kind of like a recipe from a cookbook. And that recipe is transcribed, and then it is sent from the nucleus, or if we go back to our analogy, from the library, and it's sent out to the chefs. Well, the chefs are the cooks. They make things, and they are a lot like ribosomes. Like chefs make meals from the instructions in a recipe, ribosomes are going to make protein from the instructions that are found within messenger RNA, or mRNA. So to review this by analogy, again, the DNA that we find within our nucleus is quite like the cookbook that we have here. That DNA has to be transcribed into an RNA form by RNA polymerase, and that RNA is ultimately going to be translated into an amino acid or a protein sequence, and this is carried out by the ribosome. Let's take a look again at how this occurs within the cell. Now, again, what we're going to do like we did in the past is we're going to label this inner structure here as the nucleus. And so everything on the inside of this quarter circle here is happening inside the nucleus, and everything outside of it is taking place in the cytoplasm. We talked about how within the nucleus we have double-stranded DNA. And so we're going to go ahead and we're going to label part of that. We're going to put a little bubble here in the middle. And in that bubble, the DNA is going to be single-stranded or separated from each other. And we want to make sure real quick to label our 5' and 3' ends of this DNA. We talked about how at this structure here, we will find a section of the DNA known as a gene. Genes code for, ultimately, proteins. And within this gene coding region, we might find this little enzyme right here, which is our RNA polymerase. And I'm just going to abbreviate that for the time being. We said that the RNA polymerase has the ability to separate DNA into single strands, and it reads a template DNA, and as it's doing so, it's making the RNA complementary to that template DNA. And that RNA kind of exits through a little tunnel here from the inside of that particular section of DNA. So this would be the 5' end of our mRNA here. Here are all the nucleotides shown as hash marks, and where it's still binding to the DNA. Do recall that this region, or this part of the DNA right here, the one that is being read by the RNA polymerase, is the template DNA. template DNA and mRNA are antiparallel and complementary to one another. After the mRNA is made, it is exported from the nucleus through nuclear pores, and it makes its way into the cytoplasm. Within the cytoplasm, it's going to encounter ribosomes. So what we're going to do is we're going to draw our mRNA right here. Make sure to, again, kind of show these lines here representing our nucleotides, or the nitrogenous base of our nucleotides. Label our 5' and 3' ends. And now we're going to draw our ribosome. The ribosome is a large two-part structure consisting of a large subunit and a small subunit, as shown here. And the ribosome moves down the mRNA in 3 nucleotide jumps. We'll talk again about the importance of these 3 nucleotides in a minute. But as the ribosome moves down, tRNAs will be bringing amino acids to the ribosome. And as those tRNAs deposit the amino acids from this ribosome here, we will have a growing polypeptide, or protein. The process of translation is always going to begin at what we call the start codon, which is this sequence right here, AUG, and it will always continue until it reaches a stop codon. And there are 3 different stop codons. You do not need to remember what they are. Let's now take a look at how DNA is converted into RNA through the process of transcription, and how that RNA is converted into an amino acid sequence through the process of translation. You'll recall that sense DNA and template DNA are anti-parallel from each other, and they are also complementary. And so ATs pair together, TAs pair together, GCs pair, as do CGs. The mRNA sequence that we're going to write below is actually complementary and anti-parallel to the template DNA. Take a minute to determine the mRNA sequence for yourself. Pause the video at this time. Note in the mRNA sequence here that it is anti-parallel to the template DNA, and we have complementary base pairs. The difference between the mRNA and the sense DNA on top is that whereas we saw T's before, we now see these replaced with U's in the mRNA sequence. Now, like before, what we're going to do is we're going to take this mRNA and divide it into 3 nucleotide chunks, as shown here. Remember that each of these 3 nucleotide chunks is known as a codon, and each codon codes for an amino acid. In order to determine the amino acid sequence that is produced by our mRNA, we need to use the codon code that is found at the bottom of this image. Recall that in reading this codon code, we read from the inside to the outside. And so in our case, the first codon in the mRNA is AUG. We would begin at the A in the middle, then proceed to the U following it, and finally finish up at the G down here at the bottom. And that tells us that AUG codes for the amino acid methionine, or MET. In addition to coding for methionine, this codon is also known as the start codon. We find this at the beginning of every single mRNA sequence that codes for a protein. Let's try this one more time. The next codon in our sequence is GAU. Again, if we work from G in the middle to A, and then finally to U, we find ourselves at the codon for aspartate. And so ASP would be the next amino acid within this sequence. At this time, pause the video and try to figure out the remainder of the amino acid sequence on your own. So once this amino acid sequence is completed, it should read MET, ASP, HIS, LU, VAL, and at the end here, UAG codes for a stop codon. And there are 3 different stop codons that are shown over in this region over the codon code. You can write stop if you'd like to, or just leave that location blank. So to finalize our conversation, let's just take a little bit of a dive, very so briefly, into how the ribosome works. I want to remind you that the form of RNA that is translated is the mRNA. The RNA that is part of the ribosome is the ribosomal RNA. And the RNA that transfers amino acids to the ribosome is the tRNA. These 3 work together in the process of translation. We've drawn already once how the ribosome is kind of this large structure here consisting of 2 units. That the mRNA, as I'm going to show it in green here, kind of fits in between those 2 units. And that the tRNAs are going to bring into these ribosomes the amino acids. And in order for those amino acids to come in the correct order, the tRNA has to pair complementary and anti-parallel to the mRNA sequence. The region where the tRNA pairs complementary and anti-parallel to the mRNA is a structure that we've already talked about, and that is the 3 nucleotide sequence known as a codon.